Our today's topic for discussion is enzymes. Enzymology is the branch of biochemistry that concerned with the study of enzymes. So in our today's lecture of enzymology, we will discuss about the introduction, classification, chemical nature and properties, factors affecting enzyme activity, coenzymes and the regulation of enzyme activity. So, what are enzymes? Enzymes are the biocatalysts that are synthesized by living cells. They are proteins in nature, colloidal and thermolabile in character, and specific in their actions. Now, the enzymes are sometimes considered under two broad categories. The first is the intracellular and the second is the extracellular enzymes. Intracellular enzymes are synthesized and retained in the cell for the use of cell itself. They are found in the cytoplasm, nucleus, mitochondria and chloroplast. Example, oxidoreductase catalyzes the biological oxidation and the enzymes involved in the reduction in the mitochondria. All these are the examples for the intracellular enzymes. The second are the extracellular enzymes, and extracellular enzymes are synthesized in the cell but secreted from the cell to work externally. Example, digestive enzymes produced by the pancreas are not used by the cells in the pancreas but are transported to the duodenum for its functioning. Enzymes are generally named according to the reaction they catalyzed or by suffixing ase, ase after the name of the substrate. For example, lactose is the substrate which is catalyzed by the enzyme lactase. Maltose is the substrate which is catalyzed by the enzyme maltase. And the cellulose is the substrate which is catalyzed by the cellulase. So you are observing that all of these enzymes name were suffixed by ase. So lactase, maltase and cellulase represents various enzymes which catalyze the substrates like lactose, maltose, cellulose respectively. The International Union of Biochemistry, that is IUB, the International Union of Biochemistry, the IUB, appointed an enzyme commission in 1961. IUB has classified the enzymes into 6 major classes in 1964. Now coming to the classification of enzymes. Enzymes can be classified into various groups like oxidoreductases, transferases, hydrolases, lyases, isomerases and ligases. All of these enzymes can be recognized by using a word othle. This is the word which can be used to learn the classification. O for the oxidoreductases, T for the transferases, H for hydrolases, L for lyases, I for isomerases and L for ligases. So coming to the first classification, that is oxidoreductases. Oxidoreductases are the enzymes which are involved in the oxidation reduction reaction. Example is lactate dehydrogenase or the alcohol dehydrogenase. The second class of the enzymes include the transferases, and these are the enzymes that catalyze the transfer of functional groups. Example is hexokinase. The next class of enzymes are hydrolases, and these are the enzymes that bring about hydrolysis of various compounds by addition of water. Example is lipase or lysozyme. The next class of enzyme are lyases, And these are the enzymes which are specialized in the cleavage of the bonds by atom elimination and resulting in double bond. Example are the fumarase or the aldolase enzyme. The next class of enzymes are the isomerases, and the isomerases are the enzymes which are involved in all the isomerization reaction. And example is triose phosphate isomerase or the retinol isomerase. Now the next class of enzymes are the ligases. These are the enzymes which can catalyze the synthetic reaction where two molecules are joined together and ATP is used. And the example is RNA polymerase or the succinate thiokinase. So, it is important to note that the suffix is added to the name of the substrate to find the trivial names of the enzymes. Now coming to the chemical nature and properties of enzymes. The first is the holoenzyme. Holoenzyme is the functional unit of enzyme which is made up of apoenzyme, that is the protein part, and the coenzyme, which is the non-protein part in the enzyme. The second is the prosthetic group. When the non-protein moiety tightly binds with the apoenzymes, in this case the coenzyme cannot be able to separate from the apoenzyme, is known as a prosthetic group. The third is monomeric enzymes. The enzymes which are made up of single polypeptide unit is known as monomeric enzymes. Example is ribonuclease and trypsin. The fourth is the oligomeric enzyme. The enzymes which possess more than one polypeptide chain are known as oligomeric enzyme. Example is lactate dehydrogenase. The fifth is the multi-enzyme complexes which can possess specific sites to catalyze different reaction in a sequence. And the example is pyruvate dehydrogenase. Now coming to the factors affecting enzyme activity. There are various important factors that can influence the velocity of enzyme reaction. The first and important factor is the concentration of the enzyme. The concentration of the enzyme, when increased, can result in the increase in the velocity of the reaction. The second is the concentration of the substrate. Increase in the substrate concentration gradually increase the velocity of the enzyme reaction within the limited range of the substrate levels. If we plot a graph for the concentration of the substrate, then this is the graph on which x-axis we will take the substrate concentration, and on y-axis we can take the velocity, and we can get a rectangular hyperbola. Now the next factor which can affect the enzyme activity is the effect of temperature. The velocity of reaction increases with the increase in temperature up to a maximum and then declines. A bell-shaped curve is observed. So if we plot the curve for the effect of temperature, then we can take temperature on the x-axis and enzyme velocity on the y-axis, and we can get a bell-shaped curve. Now next is the effect of pH. The effect of pH is same as the effect of the temperature, and here also we can get a bell-shaped curve, and the curve is plotted between the pH at the x-axis and the velocity of enzyme at the y-axis, and here also we can get a bell-shaped curve. Now the next is the effect of product concentration. The accumulation of the product generally decreases the enzyme activity. So it is point to be noted that we have to Keep in mind that we cannot accumulate the product which is obtained after the catalysis actions of enzyme. Now the next is the effect of activators. Some enzymes require certain inorganic metallic cations like Mg2 plus, Mn2 plus, Zn2 plus, Ca2 plus, etc. for their optimum activity. So if these inorganic cations are not available, so these enzymes will not give their optimum activity. Now coming to the coenzymes. The non-protein organic low molecular weight and dilyzable substances associated with the enzyme functions is known as coenzymes. And there are various important properties of these coenzymes. The first and important property is that coenzymes are the secondary substrate. That is, they are also called as co-substrate. They also undergo alteration during the enzymatic reactions, which are later regenerated. The next and important property is they are involved in the transfer of atoms or the groups like hydrogen, keto, amino, etc. Coenzymes helps in the transportation of the substrate from one point of the cell to the another. Thus serve as a recyclable shuttles. Example, UDP glucose, in which UDP is the coenzyme. And this UDP glucose will form the UDP glucose. Now the next and important property is that most of the coenzymes are vitamin B complex derivatives. Like if we see the example, the biotin is the vitamin which is used as biocytin coenzyme. Cobalamin is the vitamin which is used as cobalamin coenzyme. Folic acid is the vitamin used as tetrahydrofolate. Nicotinamide is used as nicotinamide coenzyme. Pantothenate is used as coenzyme A. Pyridoxine is used as pyridoxal phosphate. Riboflavin is used as flavin coenzyme, and thymine is used as thymine pyrophosphate. So here we are observing that most of the coenzymes are the vitamin B complex derivatives. Now there are also some non-vitamin coenzymes are also present, and some non-vitamin coenzymes are ATP, CDP, UDP, etc. Also there are some nucleotide coenzymes which contain the nucleotides like nitrogenous base, sugar and phosphate, and these are the NAD plus, NADP plus, FMN and FAT, etc. All these are the coenzymes which are nucleotide in nature. And also there are the coenzymes which are protein in nature. For example, thioredoxin. Thioredoxin is a coenzyme which is proteinous in nature. There is one also important point to be noted here, that the specificity of the enzyme is mostly dependent on the apoenzyme and not on the coenzyme. Now coming to the regulation of enzyme. There are three important types of regulation of enzyme. The first is the allosteric regulation, the second is the feedback regulation, and the third is the active and inactive enzymes. So coming to the first, that is the allosteric regulation. The non-covalent reversible binding of effector molecule at the allosteric site brings about a conformational change in the active site of the enzyme. So binding of an allosteric effector induces conformational changes in the enzyme. and this conformational change can leads to the inhibition or the activation of the catalytic activity of the enzyme. These allosteric activator and inhibitor exhibit positive and negative cooperativity with the substrate. The allosteric activator on binding to the allosteric site promote the binding of the substrate to the active site or the catalytic action. But the allosteric inhibitor has an opposite action. Now the second is the feedback inhibition or the feedback regulation. The process of inhibiting the first step by the final product. This is a real cellular economy to save the cell from the wasteful expenditure of synthesizing a compound which is already available within the cell. For example, if we consider A as an initial substrate, B, C and D are the intermediate, E is also another intermediate, and F is the end product, and the pathway is catalyzed by the different enzymes like E1, E2, E3, E4 or the E5. So in this condition, there is a feedback is given to the enzyme to stop its. So an indication is given to the first step by the final product, and feedback is given by the final product to the first enzyme to stop the reaction. So this is a feedback regulation. The third type of a regulation includes the active and inactive enzymes. The enzymes are synthesized as proenzymes or the zymogens, which undergo irreversible covalent activation to get an active enzyme. For example, chymotrypsinogen will give chymotrypsin, pepsinogen will give pepsin, plasminogen give plasmin, or the trypsinogen can give trypsin. These enzymes are interconvertible and will be activated in the body depending on the needs of the body. So these are the three major type of the regulation of the enzyme activity. The first is the allosteric regulation, in which the effector can bind on the allosteric site and will modify the action of the enzyme. I am using the term modification because some enzymes have allosteric site, that is an additional site besides the active site on the enzyme. So certain substances binds to these site and modulate the activity of the enzyme. I am using the term modulation because when a negative allosteric effector binds at this site, it will inhibit the enzyme activity. But if a positive effector will binds at this site, it will increase the enzyme activity. So this is the allosteric regulation. The second is the feedback regulation, in which the last product, the final product, which is if already present in the body, can indicate to the first enzyme not to further proceed the reaction for the product. And the third is the active and inactive enzymes, in which the enzymes are only activated when there is need of the body. So these are the three main types of the regulation of the enzymes. So this is all about enzymes. If you like this video, please do subscribe my channel. Thank you for your time.